Welcome to the SANS Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. If you're enjoying this deep dive into groundbreaking research, please remember to like, subscribe, and maybe share it with anyone curious about this stuff. Your support really does help the SANS Science Experience grow. Okay, so today we're diving deep into cellular senescence, the ones people often call zombie cells. We're looking at some really interesting new work that shakes up our understanding of how they actually function in aging and disease. Now, you've probably heard about Senolytics drugs meant to clear these cells out, but there's this persistent mystery, right? Why do some senescent cells just stick around?
That's exactly it. Cellular senescence, it's definitely got this dual nature. Sometimes it's beneficial to wound healing or stopping early cancer growth. But when these senescent cells hang around too long, they often turn harmful. They pump out inflammatory signals, damage nearby tissues. It's a big factor in aging and lots of diseases. And Senolytics, they were designed to target and eliminate these bad actors selectively. But like you said, they typically only manage to clear maybe 30 to 70%, so there's this big chunk left over. And this new research is finally starting to give us clues about why that is.
Okay, let's unpack that then. These 30 to 70% that don't get cleared by Senolytics, are they just like tougher versions, or are they fundamentally different somehow?
It turns out they have really distinct profiles. It's fascinating. The sources show that the Senolytic-sensitive cells, the ones that do get cleared, they're kind of the bad actors here. They're highly pro-inflammatory. They're more pro-apoptotic, meaning they're sort of primed to die anyway. And they release more of this damaging mitochondrial DNA, mtDNA, which really triggers inflammation. And critically, if you take these cells and transplant them into young mice, you see real physical problems, frailty, weaker grip strength.
Wow, okay, so those are clearly the ones we want to get rid of.
Exactly. Now, contrast that with the Senolytic-resistant cells. They're still senescent. Definitely, they show the usual markers like P69, K4A, P21, that sort of thing. But their senescence-associated secretory phenotype, the SASP, basically the cocktail of molecules they release, it's fundamentally different.
How so? Different how?
Well, they're much less inflammatory, less pro-apoptotic, too. Instead, their SESP is richer in things like growth factors, fibrotic factors. They release less of that mtDNA we talked about. And they even express these immune evasion signals, like a protein called GPNMB, which sort of helps them hide from the immune system. And when these resistant cells are transplanted, they cause much less physical dysfunction in the mice.
That distinction is huge. Okay, but here's where it gets really interesting for me. These aren't fixed categories, are they? This idea that the cells can actually switch between being sensitive and resistant, that feels like a total game changer.
It absolutely is. This interconvertibility is, it's a profound insight. It means we're not just dealing with static populations, but a dynamic system, one we might actually be able to nudge, maybe even reprogram. The research shows you can actually push these cells one way or the other. So, for instance, if you use something called a JAK-STAT inhibitor, Ruxolitinib, as an example, this blocks a key signaling pathway involved in inflammation. It actually dampens down that pro-inflammatory SASP. And what happens? The senescent cells become more resistant to Senolytics. They sort of hide better from the drugs.
Okay, so you can make them more resistant.
Right, but you can also go the other way, using what they're calling xenosensitizers. These are often factors that kind of mimic pathogens, things like LPS or TLR3 agonists. They're basically pathogen-associated molecular patterns, PAMPs. And what they do is they amplify the pro-inflammatory SASP. They essentially trick the resistant cells into becoming highly inflammatory and pro-apoptotic again.
Ah, so you're making them visible to the Senolytics again?
Precisely. You convert those resistant cells back into a state where the Senolytics can actually work on them.
So, okay, practically speaking, what does this mean for you, for me, for future treatments, especially for really tough conditions like certain cancers?
Well, it opens up a whole new therapeutic strategy, really. Imagine developing these xenosensitizers to specifically target those senescent cell populations that current drugs can't touch. And the sources mention some really exciting, though preliminary data, particularly in triple-negative breast cancer cells, TNBC, which are notoriously tough to treat. They tested this sort of 1.2, 0.3, 0.4 stepwise approach. First, chemo like cisplatin, then a senolytic like Dasatinib, then, crucially, a xenosensitizer, a TLR3 agonist in this case, and then another round of this senolytic Dasatinib.
Okay, so a multi-step attack.
Exactly. And the result, they saw about a 90% further reduction in the cancer cell survival compared to just the chemo and senolytic alone. This isn't just about aging anymore. It's potentially a powerful new way to tackle drug resistance in really aggressive cancers and maybe, a whole range of other diseases linked to senescence.
What an incredible deep dive. This idea that senescent cells aren't static, that they can shift identities, it's really something. It leaves you wondering, doesn't it? Could targeting these cellular identities, not just trying to kill the cells, lead us somewhere new? Maybe towards actually controlling aging itself, not just managing diseases one by one. Anyway, if you learned something new today, please do hit that like button and subscribe to the channel. That way you won't miss our next exploration. Thank you for being part of the Sun's science experience.